So good evening. I'm sorry, I didn't realize that I went live. So the session was supposed to start at 5 o'clock. I think by some mistake I went live earlier. So, so good evening all of you. If you're joining the session for the first time, this is Dr. Shanmugapriya. I'm professor and head of the Department of Biochemistry at Government Madras Medical College, and I'm your Unacademy educator for Biochemistry. And today's session is on recombinant DNA technology. I will be telling you all concepts and high yield MCQs related to this topic. And before we start the session, let me tell you a few facts about Unacademy plans. The first fact that I want to tell you all is about this month NEET PG Combat. And as you all know, Combat exam is a national level scholarship program. And this program will be conducted on a monthly basis, and for this month it is to be conducted on January 23rd, and the time is 11 a.m. And every question paper will have 60 minutes, and you will have to attempt 45 questions. And this month NEET PG Combat questions will be from medicine, dermatology, anesthesia, psychiatry and radiology. And if you happen to get one among the first 200 ranks, you get a scholarship to be a plus subscriber or to be an iconic subscriber of Unacademy. And apart from that, 50 lucky participants get to win Amazon gift vouchers from a pool of 10 lakh gift vouchers. So if you ask me, apart from the scholarship and the gift vouchers, what is more important is if you attempt these national level exams, you will clearly understand where you stand. And this combat will be covering every subject in depth. So it is exactly what you need at this point of time. Good evening, Chole Bature. Yeah, good evening. And apart from this, if you're a plus subscriber or iconic subscriber, you will be able to participate in all these structured batches which we have planned for upcoming exams. The first one is Focus FMG 2022 Comprehensive Batch. The next one is Target NEET PG 2022 MCQ Discussion Batch. And both these batches started just yesterday. FMG, it is 4 months duration, and Target NEET PG 2022, the duration is 2 months. And what is new about all these structured batches when compared to previous batches is this batches we have subject-wise tests. So as soon as every subject gets covered, or after every subject gets taught, there is also a subject-wise test by the end, and that will be followed by a detailed explanation of the test by the educator. And apart from this, grant tests are being conducted in a cyclical way. The upcoming exam, the grant test is on 22nd January at 9 a.m. for NEET PG aspirants, and on 30th January at 9 a.m. for FMG aspirants. So I have planned for an Unacademy Plus course for NEET PG aspirants on 24 January, and the duration of this course is 4 hours. The first 2 hours will be between 5:30 and 7:30 p.m. The next 2 hours will be between 8 and 10 p.m. So within 4 hours we'll be covering the entire biochemistry using high yield MCQs. That's a plan for NEET PG aspirants. And if you're an FMG aspirant, again there is a plus course with subject-wise test, and the duration of this course is 40 hours, and the course will be starting on January 27th, and it ends on Feb 5th. So on Feb 4th there will be a subject-wise test followed by a detailed discussion of the same on the last day. So that's about FMG aspirants plus course. And these are our toppers in the latest NEET PG September 2021, and these are our toppers in the latest FMG December 2021. And there is a change in the Unacademy subscription plan. Till some time back it was 3 months, which was the minimum slab. Now it is even for 2 months you get a subscription, and that would cost you 6,750. So this is a subscription plan for corresponding duration. But if you use any of the, good evening DNA, but if you use any of the educator's code, you get an additional 10% off. You can use any of my codes. One is Biochem Live or C Shanmugapriya, my name. So both these codes can work. So that's all about Unacademy plans. Let's start discussing. Duration of the session will be approximately 45 minutes DNA. Okay. So this is the first question. Which is true about recombinant DNA technology? Can anybody try to answer this? which is true about recombinant DNA technology? Does it require Taq DNA polymerase? Is it an in vitro cloning process? Does it require a vector? The equipment required is thermocycler. Can anybody answer? Can anyone who has joined the session live try to answer this? The answer is it requires a vector. Recombinant DNA technology requires a vector. So how do we exclude the other choices? It is easy to exclude the other choices because we know very well that Taq DNA polymerase and thermocycler, both these are required for polymerase chain reaction. Don't we all know that? Very good Shankar, very good Arundhati. So for polymerase chain reaction, we need Taq DNA polymerase and we need the equipment which is thermocycler. And do you think recombinant DNA technology is an in vitro cloning process? It is not an in vitro cloning process. Recombinant DNA technology is an in vivo cloning process, because in recombinant DNA technology what we do is we take the gene of interest, we attach it to a vehicle to form a recombinant vector, and we introduce that recombinant vector into E. coli. And within E. coli, amplification happens. So amplification is happening within a cell. So recombinant DNA technology is an in vivo cloning process. Do you all understand this? Yes. Whereas polymerase chain reaction, the amplification happens outside the cell. So that is an in vitro cloning process. So what is the answer, all of you? It's a self-explanatory simple question. So the answer for this would be, one second. So the answer for this would be, it requires a vector. That's a true statement. Okay. Now the next question. All the following are the tools required for recombinant DNA technology except. So this is a question on tools required for recombinant DNA technology. You know the answer already. Can you all try giving me the answer? Does it need restriction endonucleases, blotting, thermocycler, vector? You know the answer, right? What did I tell you in the last question? I said thermocycler is necessary for which technique? It is necessary for polymerase chain reaction. It is not necessary for recombinant DNA technology. So let me tell you quickly. Very good Srinidhi, very good Paramita Saha, Indu DNA. No, Indu has made a mistake. Arundhati, Shankar, good. So let's try to understand the steps involved in recombinant DNA technology. And I want you all to listen to the steps of recombinant DNA technology, imagining that we are trying to produce insulin on a large scale. And if you want to produce insulin on a large scale, what is the first raw material that you would need? Yeah, the first raw material that you would need is insulin gene. From where will you get this insulin gene? You will get this insulin gene from human chromosome. So the first step is you take human chromosome. Do you see that in the image? You take a human chromosome and you cut this human chromosome into multiple fragments like this using a DNA scissor called as restriction endonuclease. So tell me, what is the first tool that is necessary for recombinant DNA technology? It is restriction endonuclease, which cuts human chromosome into multiple fragments. And if you're asking me, why do we have to cut human chromosome? Because only then in the next step you will be able to identify the fragment with gene of interest and you will be amplifying only that fragment. Yeah, if you don't cut it, if you're going to amplify the whole human chromosome, it is a waste of time and waste of raw materials. Do you all understand this? So what do we do? The first step is we take human chromosome, we cut human chromosome into multiple fragments using DNA scissor called as restriction endonuclease. So that's the first tool that you want for recombinant DNA technology. Now in the next step, you're going to identify the one fragment which has got the gene of interest. And for that we employ a technique called as hybridization technique or blotting technique. So tell me, what is the second tool that is necessary for recombinant DNA technology? It is hybridization or blotting technique. For now, just listen to the steps. In another few minutes, I will be telling you in detail about every tool. Okay. So what is the second tool that is necessary for recombinant DNA technology? It is hybridization technique or blotting technique. So using that technique, you have identified that this is the fragment with gene of interest. Now you have to introduce this fragment with gene of interest into E. coli. For that you need a vehicle. What is a vehicle? It should carry the fragment with gene of interest and it should get into E. coli. And that vehicle is called as a vector. So tell me, what is the third tool that is necessary for recombinant DNA technology? It is a vector. So what we do next is, we are going to introduce the fragment with gene of interest into vector, for which you have to cut the vector. So again, you use the same restriction endonuclease and you cut the vector to look like this. And after you have cut it, you are going to introduce the fragment with gene of interest, okay, to form something called as recombinant vector. So now do you understand why the technique is called as recombinant DNA technology? Yeah, why do we call it as recombinant DNA technology? Because here we are linking two unrelated genomes. What are the two unrelated genomes? One is a eukaryotic gene fragment, the other one is prokaryotic vector, to form recombinant vector. That is why the technique is called as recombinant DNA technology. And after you have formed recombinant vector, now comes the most difficult step of recombinant DNA technology, wherein we are going to introduce this recombinant vector into E. coli. Okay, why am I saying it is difficult? Because you are trying to introduce such a larger particle across cell wall of E. coli. Can you see that here? You have successfully introduced the recombinant vector into E. coli, but to be able to do this, you should cross what? You should be able to cross the cell wall. So that's going to be difficult. So what we do is, using high electric currents, we introduce pores onto the cell wall. So this step is called as electroporation step. So what do we do in electroporation step? Using high electric currents, we introduce pores onto the cell wall, and through these pores the recombinant vector will be taken up. So this is the rate limiting step of recombinant DNA technology. This was once asked. So tell me, what is the rate limiting step of recombinant DNA technology? All of you? What is the rate limiting step of recombinant DNA technology? Introducing the recombinant vector into E. coli. Because the effectiveness of recombinant DNA technology will depend upon how efficiently you have introduced the recombinant vector into E. coli. And once you are done with this step, the rest E. coli takes care. Because whenever the vector replicates, you get multiple copies. And whenever the cell divides also, you get multiple copies. So this is about an outline of recombinant DNA technology. Is that clear, all of you? Yeah, so tell me, what are the steps involved in recombinant DNA technology? The first step is, you take human chromosome. The repetition is for your sake. Try to repeat it along with me. So what is the first step? You take human chromosome. You cut human chromosome into multiple fragments using a DNA scissor called as restriction endonuclease. So that is the first tool that you want. And out of all these fragments, you will identify the fragment with gene of interest using hybridization technique or blotting technique. So that is the second tool that you want. And then you have to introduce this, very good. You have to introduce this fragment into E. coli, for which you need a vehicle. And what do you call that vehicle as? The vehicle is called as the vector. So the third tool that is necessary for recombinant DNA technology is a vector. Now to introduce the fragment with gene of interest into the vector, you will be cutting the vector also using the same restriction endonuclease. And then you introduce the fragment with gene of interest to form something called as recombinant vector. Now the most difficult step is introducing recombinant vector into E. coli, for which we use high electric currents and we introduce pores onto the cell wall. So that step is called as electroporation step. And once you are done with this electroporation step, the rest E. coli takes care. Whenever the vector replicates, you get multiple copies. Whenever the cell divides also, you get multiple copies. Okay. So can we try to answer this question now? All the following are tools required for recombinant DNA technology except. I hope you don't have any doubt now. Yeah, we definitely don't need a thermocycler. When will you need a thermocycler? Only when you have to cycle between temperatures. Yeah, in polymerase chain reaction, yesterday I told you denaturation happens at a higher temperature, 95. and then annealing temperature happens at the annealing step happens at a lower temperature, and then elongation. So only when you have to cycle between temperatures you need thermocycler. So this is necessary only for PCR, whereas you need the rest for recombinant DNA technology. Good. So the next question, so we are now beginning to learn about the individual tools of recombinant DNA technology. Very good, DNA. Which is not true about sticky end producing restriction endonucleases? Try to answer this. Do they cut both the strands? They cut at palindromic sequences. HPA1 is an example. It favors formation of recombinant vector. What do you think is the right answer? The right answer here is choice A, because HPA1 is a blunt end producing restriction endonuclease. Yeah, HPA1 is a blunt end producing restriction endonuclease. So let's try to understand about restriction endonucleases. So what are restriction endonucleases? These are enzymes which cut at specific sites, and those sites are always palindromic sequences. Yeah, restriction endonucleases cut only at palindromic sequences. So can you all tell me what is palindrome in literature? What does palindrome mean? Palindrome means a word or a phrase which reads the same, yeah, which is going to be the same when read from either direction. For example, rotor, R-O-T-O-R, read it from either side, it is the same. Do you understand this? So that is palindrome usually. Now what is a palindromic sequence in genetics? Palindromic sequence is a double-stranded sequence. Listen to this carefully. It's a double-stranded sequence wherein the 3' 5' strand sequence will be the same as 5' 3' strand sequence read in the reverse direction. Do you all understand this? Palindromic sequence is a double-stranded sequence wherein the 3' 5' strand sequence will be the same as 5' 3' strand sequence read in the reverse direction. For example, have a look at this. Suppose this represents the 3' 5' strand sequence. Can you all tell me what will be the 5' 3' strand sequence for this? Write complementary sequences and tell me what will be the answer. The answer will be CCTAGG, right? One second, let me erase it. Yeah, suppose this is the 3' 5' strand sequence, and the 5' 3' strand sequence would be this. And don't you see that this 3' 5' strand sequence is nothing but the 5' 3' strand sequence when it is read in the reverse direction. So this is a palindromic sequence. And depending upon how a restriction endonuclease cuts this palindromic sequence, they are of two types. One is a sticky end producing restriction endonuclease. The other one is a blunt end producing restriction endonuclease. Very good, Thillairam. So what are the two types of restriction endonucleases? Sticky end producing enzyme and blunt end producing enzyme. So first let me tell you what is a sticky end producing restriction endonuclease. This enzyme cuts both the strands at two different sites. Instead of the enzyme cutting both the strands at the same point like this, it cuts both the strands at two different points. It is cutting the top strand here and it is cutting the bottom strand somewhere here. So that what will happen to the cut ends produced? Yeah, what will happen to the cut ends produced? This will be the cut end, right? So this is going to be one end, and this is going to be the other end. So the cut ends produced are staggered. In that case, you call it as a sticky end producing restriction endonuclease, because using the same enzyme you would have cut not only the human chromosome but also the vector. Suppose this represents the end of the fragment with gene of interest. Yeah, suppose this represents the end of fragment with gene of interest, and suppose this represents the end of the vector fragment. What will happen when you bring them both close to each other? You are bringing the fragment with gene of interest and the vector. So what will happen? Because they have hanging edges, they stick together easily. Yeah, because they have hanging edges, they stick together easily. So formation of recombinant vector is easier. If it's a sticky end producing restriction endonuclease, formation of a recombinant vector is easier. Whereas what is a blunt end producing restriction endonuclease? If it's a blunt end producing restriction endonuclease, it cuts both the strands at the same point. Can you see that here? It cuts both the strands after the third nucleotide, so that the cut ends produce a blunt. So can you tell me what will be the limitation if an enzyme is a blunt end producing enzyme? If it's a blunt end producing enzyme, even after you bring the fragment with gene of interest and the vector, because they don't have hanging edges, they do not easily stick together. So you will have to use very strong DNA ligases to ligate them. Do you all understand this? You will have to use very strong DNA ligases to ligate them. So what is the limitation if you use a blunt end producing restriction endonuclease, all of you? The limitation is formation of recombinant vector is difficult. So you will have to use very strong DNA ligases to ligate them. And that is why these days mostly we use only sticky end producing restriction endonucleases, and the only blunt end producing restriction endonuclease which is still in use is HPA1. So tell me, what is the example for blunt end producing restriction endonuclease? It is HPA1. Okay. So now read all the statements and tell me if these statements are true or false. Tell me about the first statement. It is a sticky end producing enzyme. Why break and then join again? DNA, we are breaking. I told you, right? You were there when I told you. You break human chromosome. You get multiple fragments. Out of those fragments, you get one fragment with the gene of interest. Yeah, that is one aspect. The other aspect is you are cutting the vector and you are joining the fragment with gene of interest and vector. Do you understand this? We are not joining the chromosome again. We are joining the fragment with gene of interest with the vector. Do you understand that, DNA? Okay, so read the statement here. Do they cut both the strands? True. Do they cut at palindromic sequences? Yes, they cut at palindromic sequences. HPA1 is an example. Is that true? HPA1 is not an example of sticky end producing restriction endonuclease. It's an example of blunt end, yes, joining with vector DNA. It's an example of blunt end producing restriction endonuclease. That is a false statement. It favors formation of recombinant vector. False. No, that's true. Yes, sticky end producing restriction endonuclease favors recombinant vector formation. Good, Arundhati, you're right. So the next question, I hope you all know the answer for this question. Can you all give me the answer? The rate limiting step of recombinant DNA technology is formation of recombinant vector, introduction of recombinant vector into a cell, identification of the fragment with gene of interest, and cloning of recombinant vector. What do you think is the rate limiting enzyme? I told you electroporation step. Yeah, electroporation step, or the step wherein you introduce the recombinant vector into E. coli is the rate limiting enzyme. Very good, DNA. The next question, which of the following is not true about northern blot? Okay, so we are done with the first tool, which is restriction endonucleases, and using restriction endonucleases, now you have multiple fragments. To identify the fragment with gene of interest, I said you will be using a blotting technique. So this question is about blotting technique. Can anybody try answering this? Which is not true about northern blot? cDNA is the probe. Does not involve denaturation step. Separates fragments based on length. The probe can be labeled with a radioactive substance. Yeah, answer is again B. It does involve a denaturation step. Northern blot involves a denaturation step. So let's now try to understand the steps involved in blotting technique or hybridization technique. Yeah, listen to this carefully. There has been many questions on this topic. So in any blotting technique, yeah, the first step will be to separate all the fragments based on length by electrophoresis. So what is the first step? The first step is electrophoretic separation. So what I have shown you here is usually electrophoretic separation is done on agarose gel or on a polyacrylamide gel. And what is shown here is a polyacrylamide gel, and all these are wells. And into every well you have added some fragment. Do you understand this? You have all the fragments, right? So you take all the fragments, you add it to one well. Okay, after you add it to one well, you connect this tank to an electrical supply. So close to the point of application is connected to a negatively charged electrode. The opposite side is connected to a positively charged electrode. And because all these DNAs are negatively charged, all these DNA fragments are negatively charged, they will start moving towards oppositely charged electrode. And the migration gets affected by the length. Longer fragments cannot move faster. So longer fragments will stay close to the point of application and shorter fragments move faster. Okay, so this is how they would look. After electrophoretic separation, they have got separated into multiple bands. Longer fragments stay close to the point of application and shorter fragments move faster. Okay, so after electrophoretic separation, the next step is denaturation, wherein you denature all the fragments. You make them single-stranded. Can anybody tell me why do you have to denature all the fragments and make them single-stranded? Because in the next few steps, you are going to use a radio-labeled probe. And this radio-labeled probe will be single-stranded and it will be complementary to the gene of interest. And for that single-stranded probe to hybridize, this should be single-stranded. Isn't that logical? Yeah, what is the purpose of hybridization technique or blotting technique? You are trying to identify the specific DNA fragment. How will you identify the specific DNA fragment? By using a probe. And that probe will be labeled with a radioactive substance and it will be single-stranded. And for that single-stranded probe to hybridize, these fragments have to be single-stranded. That is why what is the next step? Yes, very good Arundhati. The next step is denaturation. Okay. So after denaturation, you take this gel and you blot it on a nitrocellulose paper. Because handling a gel is very difficult. Even if you touch the gel will break. So what do? We take the gel and you blot it on a nitrocellulose paper. Do you see that here? This is the gel. You are blotting it on a nitrocellulose paper. So this step is called as blotting step. And that is why the technique is called as blotting technique. Because you are blotting the gel on a nitrocellulose paper. And then you take this nitrocellulose paper and you dip the nitrocellulose paper in a solution which has got a radio-labeled probe. Have you all seen a petri dish? Yeah, so though we think of blotting as something which is very complicated, this step happens just in a petri dish. So we take a petri dish, we fill the petri dish with a solution containing radio-labeled probe, and into this petri dish we drop the paper. So this is how it would look. So once you have dropped the paper, the radio-labeled probe will try to find complementary sequences, and once they find complementary sequences, they hybridize. Do you all understand this? Suppose they find a complementary sequence here, they hybridize here. Suppose here, they hybridize here. So what will you call this step as? You will call this step as hybridization step. And that is why the technique is called as hybridization technique, because you are allowing the probe to hybridize with the fragment with gene of interest. Okay, so this is called as hybridization step. After you give enough time for hybridization, you take out the paper, you wash away excess unbound probe. You take out the paper, you wash away excess unbound probe, and then you expose the paper to X-ray. Yeah, you expose the paper to X-ray. Because these probes are radio labeled, they will form a marking on the X-ray. So do you see that here? So this is the nitrocellulose paper which you have subjected to hybridization, and these red color, can you see red color fragments here? Yeah, these red color bands represent the bands wherein the probes have gone and hybridized. Now you expose this paper to X-ray. Because it is radio label, it will form a marking on the X-ray. So this is called as autoradiogram. Yeah, this is called as autoradiogram. So by visualizing the autoradiogram, you will know which fragment has got the gene of interest. Is that clear, all of you? I hope it is clear to all those who are attending the session. Okay, so tell me, what are the steps of hybridization technique or blotting technique? For any hybridization or blotting technique, the first step is you have to separate all fragments based on length by electrophoresis. After you separate all the fragments, you denature the fragments. You make them all single stranded. Okay. After denaturation, you take the gel and you blot it on a nitrocellulose paper. And that is why the technique is called as blotting technique. After blotting, you take the nitrocellulose paper and you dip the nitrocellulose paper in a solution containing radio-labeled probe. And these radio-labeled probe will try to find complementary sequences. Once they find complementary sequences, they hybridize. So this step is called as hybridization step. And after you give enough time for hybridization, you take out the paper, you wash away excess unbound probe. And then you expose the paper to X-ray, that's called as autoradiogram. And wherever the probes have hybridized, it will form a marking on the X-ray. So that will tell you which fragment has got the gene of interest. So tell me, what are the 6 steps involved in hybridization technique or blotting technique? Electrophoretic separation, denaturation, blotting, hybridization, washing step, and finally autoradiogram. And this technique is called as southern blotting, named after the person who invented this technique. So whenever you're trying to use this principle, whenever you're trying to use these principles or this procedure to identify an unknown DNA fragment, that's called as southern blotting, named after the person who invented this technique. If you're using the same principle, but for identifying something else, you will have other blotting techniques. So tell me, what is northern blot? Northern blot is the name that is given if you are using the technique for identifying unknown RNA. So what do you think is the difference between southern blot and northern blot, all of you? What is the difference between southern blot and northern blot? One is, I told you, southern is used for identifying DNA, northern is used for identifying RNA. But technique wise, what do you think is the difference? In southern blot, when you are trying to identify a DNA which is double stranded, there should be a very strong denaturation step. There should be a very strong denaturation step. Whereas when you are trying to identify an unknown RNA which is already single stranded, will you employ a denaturation step in northern blot, all of you? Will you employ a denaturation step? Yes, there will be a denaturation step even in northern blot, but it will be a milder denaturation step. Why will it be milder? The milder denaturation step is required to break any hairpin loops in single stranded RNAs. Though RNAs are single stranded, anything which is single stranded will have hairpin loops. And until there are hairpin loops, they won't be able to hybridize with the probe. So what are you supposed to do? Even in northern blot, there is a denaturation step, but it is a milder denaturation step. Do you understand this? And if you are using the same principle, but for identifying unknown protein, that is called as western blot. To identify an unknown protein, the technique is called as western blot. So what do you think is the difference between southern and northern blot on one hand and western blot on the other hand? What do you think is the difference? Southern and northern blot on one hand and western blot on the other hand. What is the probe that is used in southern and northern blot? Be it a DNA identification process or an RNA identification process, the probe that is used will be a DNA that is complementary to them. So what is the probe that is used in southern and northern blot? In southern and northern blot, we use a cDNA probe. Whereas in western blot, when we are trying to identify a protein, you cannot use a DNA probe. What should be used as a probe? You have to synthesize monoclonal or polyclonal antibodies directed against these proteins, and then you can use them as a probe. So what is the probe that is used in western blot? This has been asked so many times in NEET. So what is the probe that is used in western blot? It is antibodies. Very good, Shankar. Okay, so that is about differences between all these blotting techniques. So can you try answering this? Which of the following is not true about northern blot? Does it involve cDNA as the probe? Can you all tell me, true or false? Does it involve cDNA as the probe? Yes, true. Does not involve denaturation step? False. Even in northern blot, there is a denaturation step, but it is a milder denaturation step. So this is a false statement. Does it separate fragments based on length? Be it southern blot or northern blot, you're going to separate them based on length. So this is true. The probe can be labeled with a radioactive substance. Yes, true. Okay. So the false statement here is it does not involve denaturation step. The most commonly used plasmid vector is, so we have learnt about restriction endonucleases, we have learnt about hybridization technique or blotting technique. So now we are going to learn about vector. So the most commonly used plasmid vector is, this was asked in NEET, exactly the same question was asked in NEET. So what do you think will be the right answer? The right answer is PBR 322. The most commonly used plasmid vector is PBR. Very good Srinidhi, very good Arundhati. It is PBR 322. So let me tell you what are the qualities you expect in a vector. Because you cannot just like that take an oligonucleotide or a polynucleotide and cannot ask it to act as a vector. There are few qualities that they should exhibit. So what are the qualities a vector should exhibit? One is the vector should not be a passive vehicle. It should not just carry the fragment with gene of interest. It should not just get into E. coli and remain dormant there. Even after it gets into E. coli, it should be active in the sense it should be taking part in replication. Only then you will get multiple copies, right? So what should be the quality a vector exhibits? The vector should be capable of self-replication. Yeah, that is very important. The vector should be capable of self-replication. Or in other words, they should have one or more origins of replication. Yeah, they should have multiple origins of replication. That is the first quality. Second quality is they should have the restriction site of the enzyme that has been chosen. Yeah, they should have the restriction site of the enzyme that has been chosen. So first tell me, how do you choose a restriction enzyme? You choose a restriction enzyme in such a way that it does not cut the gene of interest. Isn't that logical? Yeah, what will happen if you use a restriction enzyme which cuts right in the middle of insulin gene? You will not get insulin products, right? So you choose restriction enzyme in such a way that it does not cut the gene of interest. Having chosen the restriction enzyme, then you choose the vector in such a way that it has got that particular restriction site. So the second quality that the vector should exhibit is it should have restriction site of the enzyme that has been chosen. Third quality is it should have one or more marker genes. Yeah, the third quality is it should have one or more marker genes. So if you're asking why will we need a marker gene, it is because, as I told you, formation of recombinant vector is not that easy. Yes or no? Formation of recombinant vector is not that easy because you are trying to link two unrelated genomes. One is a prokaryotic vector, the other one is eukaryotic gene fragment. So formation of recombinant vector is not that easy. And even after you have formed a recombinant vector, introducing this recombinant vector into E. coli is again difficult. So after you make an attempt to introduce a recombinant vector into E. coli, there should be a way of you identifying those E. coli colonies which have taken up the recombinant vector. Isn't that right? Isn't that logical? Yeah, after you have made an attempt to introduce the recombinant vector into E. coli, there should be a way of you identifying those E. coli colonies which have taken up the recombinant vector. Otherwise, what will happen? Otherwise, in a colony, so like what I told you about hybridization technique, which sounds complicated, even cloning or recombinant DNA technology might make you think of it as a complex process. But where do you allow cloning and amplification to happen? On a colony culture plate. Yeah, have you all seen a colony culture plate? You must have, right? So just on a colony culture plate, we are going to do this cloning. So within a culture plate, there will be competition among E. coli colonies. So after you find out those E. coli colonies which have the recombinant vector, you should take them and you should allow them to grow somewhere else. Otherwise, those E. coli colonies which do not have recombinant vector will overgrow this. So to identify E. coli colonies with recombinant vector, these vectors should have marker genes. And if it's a natural vector that you choose, if it's a natural vector like plasmid or phage or cosmids, you choose these natural vectors in such a way that they have at least 2 resistance genes. One is ampicillin resistance gene, the other one is tetracycline resistance gene. I'm just giving you an example. So if it's a natural vector, you choose these natural vectors in such a way that they have at least 2 resistance genes. And if it's an artificial vector, you know we have artificial vectors, right? Bacterial artificial chromosome and yeast artificial chromosome. So if you're using an artificial vector like BAC or YAC, you introduce any marker gene that is easier for identification. So that way, commonly used or widely used marker gene in all artificial vectors is beta galactosidase gene. So tell me, what is the marker gene that is often used? What is the marker gene that is used in artificial vectors? In artificial vectors, it is beta galactosidase gene. So that's about marker genes. Okay, so what are the qualities a vector should exhibit? Can you all give me the answer? What are the 3 qualities a vector should exhibit? The vector should not be a passive vehicle. It should be capable of self-replication. So there should be 1 or more origins of replication. Okay, second, they should have the restriction site that has been already chosen. Third, they should have 1 or more marker genes. Natural vectors have antibiotic resistance genes as marker genes. Artificial vectors like bacterial artificial chromosome and yeast artificial chromosome, we use beta galactosidase gene as the marker gene. Okay. Now, based on the length of the genome to be amplified, we have many choices of vectors. If the length of the genome to be amplified is between 6 and 10 kilobase pairs, if the length of the genome to be amplified is between 6 and 10 kilobase pairs, we use plasmid as the vector. Okay. So, what are plasmids, all of you? Plasmids are small, circular, extra chromosomal DNA. You know that, right? What are plasmids? Plasmids are small, circular, extra chromosomal DNA. And what is the advantage of using a plasmid as a vector? Both the advantage and disadvantage of using a plasmid as a vector will be based on its smaller size. Why is it advantageous if you are using a smaller vector? Introducing the recombinant vector into E. coli is easier if you are using a plasmid vector because it's small. Introducing the recombinant vector into E. coli is easier. So many a times we use plasmids as vector. If you are using plasmid as a vector, we know we don't need the sophisticated electroporation step. Many labs do not have this sophisticated electroporation step. Still they are involved in cloning because they are going to use plasmid as the vector. Okay, so if you are using plasmid as a vector, all that you will have to do is you will have to add calcium to the medium and you will have to freeze it. You add calcium to the medium and just keep the petri dish in a freezer. That is enough. So that will introduce spores. Through those spores, plasmid vectors will be taken up. So that's the advantage of using plasmid as a vector. And the disadvantage of using plasmid as a vector is again its smaller size. Because it's smaller, you cannot amplify if the length of the genome to be amplified is more than 10 kilobase pairs. And if you are asked which is the most commonly used plasmid vector, your answer should be PBR322 because it has got 3 origins of replication. It has got ampicillin resistance gene and tetracycline resistance gene, and it has got multiple restriction sites. And that is why the most commonly used plasmid vector is PBR322. Okay. And if the length of the genome to be amplified is between 10 and 20 kilobase pairs, if it's between 10 and 20 kilobase pairs, we use phage DNA. And what do you think is the advantage of using a phage DNA? It's self-explanatory. What is a bacteriophage? It's a virus which can infect a bacteria. Okay, so if you can pack, the fragment with gene of interest into phage DNA, then the rest phage DNA will take care because this phage has got an inherent ability to inject its genome into bacteria. You know that, right? Phage has got an inherent ability to inject its genome into bacteria. So even here you do not need the sophisticated electroporation step. everything is taken care by phage DNA. If it's between 20 and 50 kilobase pairs, between 20 and 50 kilobase pairs, we use cosmids. Okay, so what are cosmids? Cosmids are hybrid plasmids. Okay, what are cosmids? They are hybrid plasmids which have cos sequences. What are cos sequences? These are the sequences which are responsible in phage DNA for packaging and for integration. Do you understand this? These sequences are responsible for packaging the genome and for integrating the genome. So if you can use a plasmid hybrid wherein it has got all the features of plasmid that is attached to cos sequences, then integrating it into the host is easier. So that's about cosmids. It is used when the length of the genome to be amplified is between 20 and 50 kilobase pairs. And most of the human genes, size will be more than 50 kilobase pairs, in which case you will have to use either bacterial artificial chromosome or yeast artificial chromosome. Particularly if the length of the genome to be amplified is more than 500 kilobase pairs, no other go, you will have to use only yeast artificial chromosome. Is that clear? So this is about choices of vectors that we have. So what are the 5 choices of vectors we have? You can use plasmids, you can use phage DNA, you can use cosmids, bacterial artificial chromosome and yeast artificial chromosome. If the length of the genome to be amplified is more than 50 kilobase pairs, you will have to be dependent on only artificial vectors. More than 500 kilobase pairs, only yeast artificial chromosome can be used. Okay. So can you give me the answer now? The most commonly used plasmid vector is, what is answer? Now it is PBR 322. This is the last question for this session. To produce insulin on a large scale, what is introduced into E. coli? Yeah, to produce insulin on a large scale, what is introduced into E. coli? Insulin gene, insulin mRNA, insulin cDNA, insulin tRNA. DNA says it's choice A. Yeah, DNA says it's choice A. Anyone else? Does anyone else want to answer this? Insulin mRNA, insulin cDNA, insulin tRNA. To know the answer to this question, okay, answer is insulin cDNA. Yeah, what should be introduced into E. coli? It is insulin cDNA. Yes. So to know why, try to answer me this question. The question to you is, do you think human genes can be cloned and expressed in E. coli? Do you think human genes can be cloned and expressed? Arundhati, answer is C. Okay, insulin cDNA. So now answer me this question. Can human genes be cloned and expressed in E. coli? Yes or no? It is partly yes and partly no. Yes, DNA you chose C. Okay. So it is partly yes, partly no. In the sense you can clone human genes in E. coli, but you cannot express human genes in E. coli. Yeah, so it is partly yes and partly no. If you are asking me why, the reason is prokaryotes do not have introns. Don't you all know that? Reason is prokaryotes do not have introns. So prokaryotes like E. coli do not have splicing machinery. Yeah, they do not have splicing machinery. So if you just like that take insulin gene, and if you introduce that insulin gene into E. coli will be able to produce multiple copies of the insulin gene. It can clone the insulin gene multiple times. But when the question of expression comes, in E. coli, all that you can do is you can convert insulin gene into something which is analogous to a primary transcript. Because introns cannot be removed, you will not be able to convert these primary transcripts into functional mRNAs. Yeah, you will not be able to convert them into functional mRNAs. So this cannot be expressed in E. coli. So what logically should you do? What logically should you do to overcome this limitation, because we are trying to do it in a prokaryote? You should take insulin gene, you should remove all introns, you should join all exons and then introduce that into E. coli. Isn't that logical? Yeah, to be able to overcome this, what we are supposed to do is you should take insulin gene, you should remove all introns, you join all exons and then introduce that into E. coli, which will be cloned and expressed. but we have not reached that far. Yeah, genetics has not reached that far because we don't still have in vitro splicing machinery. Yeah, we still do not have in vitro splicing machinery. So we are going to use in vivo splicing machinery. So what we do here is, if you are trying to produce insulin on a large scale, we maintain pancreatic beta cell culture. Yeah, they need only exons. You are right, Arundhati. So what we do is, to produce insulin on a large scale, we maintain pancreatic beta cell culture. From beta cell culture, you know, beta cells have an inherent ability to splice out introns of insulin gene to form functional mRNAs of insulin. So from beta cell culture, we extract functional mRNAs of insulin or spliced mRNAs of insulin. And then we use these mRNAs and reverse transcriptase enzyme. Yes, yeah, Nobel Prize can be obtained. You're right, DNA. So you can use reverse transcriptase enzyme. What is transcription, all of you? Transcription is DNA to RNA. So what is reverse transcription? Reverse transcription is RNA to DNA. So when you use reverse transcriptase, this reverse transcriptase will use the functional mRNA as the template and it will synthesize a single-stranded DNA first. Then it uses a single-stranded DNA as a template and it will synthesize the other strand. Such a double-stranded DNA, such a double-stranded DNA that is complementary to fully spliced mRNA is called the cDNA. This is called the cDNA. So can you all tell me what is cDNA? Whenever you asked what is cDNA, it is not sufficient if you just say it's complementary DNA. You will have to say what is it complementary to. cDNA is complementary to fully spliced mRNAs. So cDNAs do not have introns. Do you all understand this? cDNAs do not have introns. So then these cDNAs will be coupled to the vector and that will be introduced into E. coli. And this will be cloned within E. coli and will be expressed from E. coli. Is that clear? So these are the steps involved in recombinant DNA technology to produce insulin on a large scale. Do you all understand this? So what is the first step? The first step is you maintain cell culture. You extract mRNA, you extract functional mRNAs from beta cells. You use reverse transcriptase, convert those mRNAs into cDNAs. You attach those cDNAs to vector and that recombinant vector is introduced into E. coli and within E. coli that will be cloned and expressed. Okay. So that is about today's session. So what will you get from pancreatic beta cell? It is spliced mRNA. What will you introduce into E. coli? It is cDNA. Okay. So any doubts in recombinant DNA technology? So with this, today's session is over. So thank you all. See you sometime later.